Hello again, everyone. This week we're going to be talking about carbohydrates and lipids. In two different lectures, I'm going to introduce carbohydrates and lipids, and then the following weeks you're going to learn about carbohydrate metabolism and lipid metabolism. So as always, my email address is here if you need to get a hold of me. Just by way of an outline for this lecture, I'm going to just give a brief introduction, talk first about simple carbohydrates, what we mean by chirality, specifically when we talk about carbohydrates, what tautomerization is, and then we're going to talk a little bit about glycosidic bonds and polysaccharides. Carbohydrates and lipids are a major source of energy, as all of you probably know. They're stored in the body in the form of energy reserves, glycogen for carbohydrates and triglycerides for fats. And how those forms and how those are formed and how they're metabolized will be talked about in later lectures. Carbohydrates may also be bound to proteins and have important structural and regulatory functions, which we'll also be talking about. But today's lecture and the next lecture in this series are really just to introduce carbohydrates, their structures, and a very overall view of them. Carbohydrates and lipids are involved in many biochemical pathways. You're going to be talking about the TCA cycle here in about a week or two. Phospholipids are a pathway that we're going to be talking about, even the formation of glycoproteins, lipid proteins, hormones. So all these things we're going to be touched on. So it's important to have a solid foundation of what carbohydrates and lipids are. First, we'll talk about carbohydrates. So they are the most abundant compounds in nature. Cellulose, in fact, is the most abundant organic polymer on Earth. Its structure is shown here. So cellulose is found in plant cell walls. And again, it's the most abundant organic polymer on Earth. In general, carbohydrates functions in several different ways. They can serve as biosynthetic intermediates. They can provide decoration to various compounds. For example, you can have a simple sugar plus another compound. They can form vitamins, antibiotics, etc. As we just talked about, they can be structural components in plants such as cellulose, as well as cell walls in microorganisms. They can participate in biological transport, cell recognition, activation of growth factors, modulating of the immune system, and again, we'll touch on some of these as we move through these lectures. They can, as we mentioned, sugars can be decorated on proteins. This usually occurs as a post-translational modification, so the enzyme is directed within the endoplasmic reticulum. So it's usually a specific enzyme adds a specific decoration to another protein. Proteins can either contain O-linked oligosaccharides or one simple sugar, either through serine or threonine, or they can be N-linked through the amide nitrogen of asparagine. A lot of times these are required for proper folding of the protein, so that decoration of that sugar enables the protein to fold correctly. They can confer stability, and they can be involved in cell-cell adhesion. For example, some of the things that occur in the immune system and are due to these sugar decoration. Let's first talk about simple carbohydrates, though. Let's back up to the basic building block. The simplest carbohydrates have 3 carbon sugars, or they're otherwise known as trioses. So these are glyceraldehyde and dihydroxyacetone. So they have 2 hydroxyl groups. The glyceraldehyde is referred to as the aldose group. This compound has an aldehyde group at one end. So aldose has an aldehyde group at one end. So for example, glucose. Dihydroxyacetone is a ketose, and so the ketose usually has there's usually a keto group at the C2 position. For example, fructose. So glyceraldehyde is shown here. So there's the aldehyde group at one end. Dihydroxyacetone has a keto group at the C2 position. And so these are the 3 carbon sugars shown here. But as you get longer and longer carbon chains, these names still hold true. It's an aldose if it has this aldehyde at one end, or a ketose if it has this a keto group, usually at the C2 position. The carbohydrate is classified by the number of sugar units. So monosaccharides are, trioses, tetroses, hexoses. Most of these are straight chain, and we'll talk about monosaccharides. These are the basic building blocks to build oligosaccharides. So when you put a couple of these together, up to 9 or 10 sugars, usually refer to these as oligosaccharides. The most important of these being the disaccharides, and we'll talk about some of the most important ones as we move forward. And as you get bigger and bigger polymers, these are called polysaccharides or glycans, whereas the homopolysaccharides contain a single sugar, for example, all glucose units, whereas heteropolysaccharides contain more than one sugar, a glucose plus fructose repeating unit, for example. These are considered complex carbohydrates because they can get pretty crazy structures, and we'll see a couple of examples of these. So again, they're classified, monosaccharides are classified by the length of the carbon chain. So if you have 3 carbons, it's a triose, pretty straightforward. If you move up, most of the sugars are 6 carbon sugars or hexoses. So for example, glucose and fructose. Some of the 5 carbon sugars are important that are found in RNA and DNA, and we'll touch on those. You can go up to 9 carbon units and above for simple sugars. So let's talk about a couple of the important sugars. As I just mentioned, ribose is a pentose, a 5 carbon sugar, and 2-deoxyribose. So these are found in RNA and DNA, building blocks for RNA and DNA. Glucuronic acid is another example. It's an oxidation of C6 of glucose. You can see some of those structures here. Again, I'm not big on memorizing structures, but I think it is important to know that ribose and deoxyribose are building blocks of RNA and DNA, and that they are 5 carbon sugars. Some of the other carbohydrates that are found in human tissues, glucose 6-phosphate is an intermediate in glucose metabolism, which we'll learn about, and sorbitol, which is a polyol formed on the reduction of glucose, which is shown here. So this is just to give you a couple of examples of human tissue carbohydrates. And the previous examples do not contain amino groups, but there are also important amino sugars that are found in human tissues. Glucosamine being one of the most important of those. It can either be with or without an acyl group. If it has an acetyl group, if it has an acetyl group, it's an acetylglucosamine. These are common O-linked glycosylations of serine or threonine residues. So these will be glycosylated. Some proteins will be glycosylated, with these sugars. Another example of amino sugar is sialic acid. You'll see this as a terminal residue on oligosaccharide chains of glycoproteins, and it imparts a negative charge on the compound. That's just an introduction to some of the simple monosaccharides. Let's talk a little bit about chirality and what that means. So the numbering of a carbon begins from the end containing the aldehyde or the ketone group. So we talked about that a few slides ago. So it's at the top here in this diagram. Whether or not it's D or L is based on the configuration around the highest numbered asymmetric center. So in the case of D-glucose, you have 6 carbons, but the 6th carbon is not asymmetric. So there's, an OH, an H, and an H coming out. And if you were to look at this in a mirror, it would look the same. So it's not an asymmetric center. Whereas the 5th carbon would be the highest numbered carbon of asymmetric center. So if the hydroxyl group points to the right, then is defined as a D. So this would be D-glucose because the hydroxyl is at the right, whereas the other one would be L-glucose. So as you can see, the D and L sugars are mirror images of one another, or they're known as enantiomers. So if you take a mirror and put it in between the D and L glucose, they're exact reflections of each other. Whereas other stereoisomers that have the same overall structure or same overall chemical makeup, they have unique chemical names, for example, mannose, galactose, and fructose. So they're all 6-carbon um, 6-carbon sugars, but they have different names. The number of stereoisomers that you can have is equal to 2 to the n, where n equals the number of asymmetric centers. So let's start with glyceraldehyde, a 3-carbon sugar. So it just has one asymmetric center. So 2 to the 1 equals 2, so it just has 2 stereoisomers. Shown in this diagram are just the D's. You can imagine a mirror plane here and writing all the L's over here. So D-glyceraldehyde just has 2 stereoisomers, and then you can see the examples as you move down. C6 aldoses, for example, the stereoisomers of glucose have 4 asymmetric centers. So the 4 carbons in red here. So they would have 2 to the 4th, or a total of 16 stereoisomers. Again, the 8 Ds are shown here, and then the exact mirror images would show the other 8. Nearly all sugars in the body have the D configuration. So this is opposite to amino acids, which we talked about last time, which have the L conformation. And of the 8 D-aldohexoses, only 3 of these are found in significant amounts in the body. So the three of the 8 that were shown on the previous slide. And those are glucose, which is obviously your blood sugar level, your blood sugar. This is the most important of these carbohydrates. Mannose is another important one, and galactose. Fructose is the only D-ketohexose present in significant concentrations in our diet or in the body itself. So when you have different versions of a similar carbohydrate, these differences in structures are responsible for variations in their properties. That can be physical properties, such as crystalline form, solubility, how they rotate polarized light, the chemistry, how reactive they are, and the physiological properties. So how much nutrition do they have, sweetness, absorption into the body, and we'll touch on some examples of these. First, let's introduce tautomerization. So far, I've shown these all in the linear form, but in reality, in biology, these exist typically in ring forms at equilibrium, mostly in ring forms. So tautomerization is going essentially from the linear form to the ring form. So you can imagine a hydroxyl group from the last asymmetric carbon on D-glucose attacking the aldehyde here to form this ring structure. This chemistry is pretty simple. It's because aldehydes and ketones react with alcohols to form hemiacetals and hemiketals respectively. So if you have an aldehyde plus an alcohol, again you get this hydroxyl attacking the carbon on the aldehyde to get the hemiacetal. And then an alcohol can also attack a ketone in the same way to get a hemiketal. So this is essentially the chemistry that's going on for tautomerization. For example, let's talk about the glucose hemiacetal and tautomerization. So D-glucose can cyclize to form a pyranose or furanose structure. So shown here is the furanose form. where you have a 5-membered ring, whereas the pyranose form has the 6-membered ring. When you perform this cyclization, you get a new asymmetric center at C1, C1 here. So you can get two stereoisomers that can form either the alpha form or the beta form. So for example, the alpha form, the hydroxyl is pointing down, and the beta form, the hydroxyl is pointing up. Why do we care so much about the glucose hemiacetal? Glucose exists largely in this non-reactive inert cyclic hemiacetal conformations. So when we think about glucose in our bodies, it's 99.99% in this cyclic hemiacetal conformational form. Of all the D sugars in the world, D-glucose exists to the greatest extent in these cyclic conformations because it can't be oxidized very well in this conformation. It is very unreactive. So that's good because we had a lot of glucose in our body. And if it was very reactive, then it would be reacting with proteins and other things. So people believe that the relative chemical inertness of glucose is the reason that it's been selected as our main blood sugar. I've thrown some different types of structures at you just to orient you a little bit more. There's the Fisher representation, which is a linear representation, which I've shown. The Hallworth is a cyclic representation, and the ratio of these can shift as a function of temperature, pH, salt concentration, etc. So the glucose, in our bodies mostly is in this form, but you can shift it to the linear form and other cyclic forms based on the pH, salt, and temperature. You also see the cyclic forms shown in this chair conformation sometimes. This is more stereochemically correct, but again, I'm not going to make you draw structures here, but just wanted to orient you into the different forms that these can be drawn. Those are the simple monosaccharides. Let's talk through that simple monosaccharide as a building block to make larger sugars. To do that, we have to form a glycosidic bond. So carbohydrates couple to each other by a glycosidic bond to form disaccharides, trisaccharides, polysaccharides. The anomeric hydroxyl and the hydroxyl of another sugar covalently bond to liberate water. So for example, in this generic reaction here, you get the ROR forming and releasing H2O. So a very simple example for D-glucopyranose, if you add methanol, you release water, and you form this methyl group here. So you can imagine a sugar being replaced with this methanol to form a disaccharide down here in the same type of chemical reaction. The most common disaccharides are sucrose, which is table sugar. When you buy sugar, it's commercially obtained from sugarcane or sugar beet, and when you hydrolyze this, the glycosidic bond, the two building blocks are glucose and fructose. Maltose is another important disaccharide. It's malt sugar. So you get, when you hydrolyze, two molecules of D-glucose. So D-glucose is the building block for maltose. Lactose is another example, and the hydrolysis there yields glucose and galactose. Those are the disaccharides. As you continue to add more and more monosaccharides to build polysaccharides or glycans. Again, you can have homoglycans or heteroglycans. Some notable examples that we'll talk about of homoglycans are starch, cellulose, and glycogen. And the heteroglycans can get pretty complex. There are things like gums, and we'll talk about a few of those. The characteristics of these, they can have pretty high molecular weights, up to 200 kilodaltons. They can be white and amorphous products. They're not sweet. They're non-reducing, so they don't give the typical aldose or ketose reactions, and they form colloidal solutions or suspensions. Let's talk about the most important ones, amylose and cellulose. So amylose and cellulose are both built from D-glucose, but the differences in their anomeric linkage define differences in both their biochemistry and nutrition. So amylose, which is a component of starch, is an alpha-1,4 linked linear glucan. So it has alpha-1,4 linkage of glucose. Cellulose is a beta-1,4 linkage. So the hydroxyl goes up here and you get a beta linkage. So they're both D-glucose building blocks, but they have different structure. And we think about the word fiber. Fiber is actually cellulose and other non-starch polysaccharides. But this slight difference in this linkage leads to drastically different properties. So starch, or amylose, is soluble in water, whereas cellulose is not. Amylose has this pasty characteristics, where cellulose is more fibrous in nature. Amylose we can digest, you get a lot of calories from starch, whereas cellulose you cannot digest, so there's no calorie value there. And again, it's all just due to this simple difference in linkage. Starch is actually a little bit more complex than just amylose. It's amylose and amylopectin. So most common storage polysaccharide, it's the most common storage polysaccharides in plants. The glucose storage in polymeric form minimizes osmotic effects. So amylose is just this 1,4 linkage, whereas amylopectin can add in a 6,1 linkage as well. So starch itself is a mixture of these in reality. When people tried to figure out the structure of these things, they used iodine, and so when you add iodine to starch, it adopts starch itself adopts a helical formation in water, and when you add iodine to this, it inserts in this helix, and it gives a blue color. Whereas when you do this with cellulose, you don't get a reaction with iodine because you have the beta 1,4 link glucose unit. So this difference in linkage leads to differences in chemical structure. Let's talk about glycogen. So glycogen is also a glucose homoglycan. It's basically the animal equivalent to starch, but it has more alpha-1,6 branches than starch, and this permits rapid glucose release during exercise. Glycogen is stored in muscles and liver cells as high molecular weight granules. It does react with iodine, but it gives us a slightly different color, a red-violet color, because of these greater number of alpha-1,4 or alpha-1,6 branches. Glycogen is hydrolyzed by amylases that can be found in our saliva, as well as other parts of our body, as well as other enzymes to break down glycogen for calories. Those are the simpler homopolymers. There's also some more complex. Dextrans are glucose homopolymers. They have the alpha-1,4, alpha-1,6, and alpha-1,3 linkages. These have been used in shock treatments, but they've also used in gel filtration columns that we've we talked about last week. So for chromatography of proteins, the resins are often made out of dextrans. They're also a big component of dental plaques. So the dextrans can lead to formation of biofilms of bacteria. So this can lead to dental caries or tooth decay. So breakdown of teeth is caused by bacteria, typically the Streptococcus mutans bacteria. Again, this can create biofilms or dental plaques. So it's this mass of bacteria as well as these sticky dextrans. And they shield teeth from saliva in the tongue, so it makes it can cause the breakdown of teeth. Bacteria themselves ferment sugar to lactic acid, and they can remain metabolically active under these lactic acid conditions or low pH conditions. And they can feed off the sugar that you get from your diet and basically form this biofilm and lead to tooth decay. Whereas artificial sweeteners can't feed these bacteria, so they are not cariogenic. Another homoglycan of importance is chitin. So instead of glucose, this is N-acetylglucosamine homoglycan. So you get the N-acetyl group here. This is the second most abundant carbohydrate in nature. It's present in the cell wall, excuse me, of fungal organisms as well as the exoskeletons on things like lobsters and other crustaceans and some types of insects as well. It's important also because it's used commercially for coatings, so it extends the shelf life of fruits and meats and pharmaceuticals as well as medical products. Let's talk about a few examples of heteroglycans. Glycosaminoglycan is a repeating disaccharide of glucuronic acid and N-acetylglucosamine. So this is found widely through connective epithelial tissue and neural tissues. It's also a component of bacterial capsules where it plays a role in virulence. Some other heteroglycans which are important, pectins. So these are heteropolysaccharides found in various fruits such as citrus, apples, citrus fruits and apples. It's also used as jelly agents, for example, in jellies and jams. Gums are also obviously widely found in food and pharmaceutical industries. They're used as thickening agents, um, stabilizers, binding agents, adhesives. One notable example is xanthan gum. Again, these are super complex structures. No need to memorize the structures, but what's more important are knowing that these pectins and gums are heteroglycans. Let's talk about one of the most important ones, peptidoglycan. It's found in bacterial cell walls. It's both a polypeptide and a polysaccharide. So it has a polysaccharide unit and a polypeptide unit. For gram staining for the term gram-negative and gram-positive bacteria, this is due to the peptidoglycan cell wall. So you got different cross-links and thicknesses that lead to either a gram-negative stain or a gram-positive stain. So this is how bacteria are classified at the very highest level due to this peptidoglycan layer. Another important example is lipopolysaccharide. So this has a lipid anchor and a long polysaccharide. This is otherwise known as bacterial endotoxin. So it has this core oligosaccharide and then a long O antigen side chain, and again this lipid anchor. And when you have bacterial septic shock, a lot of it's due to our immune reaction to lipopolysaccharide. So this binds to toll-like receptors in our body, and it leads to a cytokine storm, essentially, and can lead to septic shock. Just another summary of some of these other glycolipids. So things that contain both a lipid component and a saccharide component. Cerebrosides, these are one sugar molecules that can found in either neurons or elsewhere in the body. You'll see things called like sulfatides. These have sulfur acid esters of some of these other cerebrosides. You'll see things called globosides, so these are ceramide oligosaccharides, and gangliosides as well. These have more complex oligosaccharides attached. They have different cell-cell recognition properties, and there can be receptors for hormones. Just to give some other examples of glycolipids, but this slide isn't terribly important to memorize. I mentioned artificial sweeteners. I'm just going to leave you guys with this as well. So artificial sweeteners, can't be digested by the body, so they don't have any calories. But just to give you some structures to show some of these that they are very close to sugars, whereas others aren't. So Splenda is actually a chlorinated sucrose, so it has just like sucrose, except for it has chlorines. It has chlorines decorated in it, and our body can't metabolize that. So you can see it's very close to sucrose, so it has a sweetness taste to it. Equal packets, as you can see here, are the chemical is actually aspartame. It's a methyl ester of aspartic acid and phenylalanine, so it doesn't really even look like a sugar. Sweet and low, you can see the structure there. Truvia is actually extracts of stevia leaf, and you can see the structure there. So I thought these were just kind of interesting to leave you guys with. And so that was the last slide. The other lecture, I'm going to talk a little bit more about carbohydrates and lipids to set you guys up for carbohydrate metabolism and lipid metabolism. And then at the end of the next lecture, there'll be a study guide as well. So I'll see you next time.